So let's concentrate now on these nucleotides that just gave everybody a lot of extra credits. You gotta love them, right? So you have the adenine, the guanine, the cytosine, and the thymine. You know that you have a five carbon sugar, and you have the deoxyribose. You also have that phosphate attached to that. Yes, everybody got in the mind the picture of the house with the swimming pool and the base that is the garage. So you have the house with the garage and swimming pool. You got your structure for your nucleotide. A strategy for enhancing memory. Please do not put in the midterm a little house with a pool in the backyard. Now, just keep the idea in your mind, okay? This is a very good study strategy, especially when you don't have a lot of time. Okay, you associate with either something that is fun or with something that is ridiculous. Okay, so you can remember it quickly. Just know the overall structure. 2 rings, purine, which one they are? Guanine, adenine. The Puga 2 business. And whatever is left is a pyrimidine, and whatever is left, what is that? Thymine and cytosine. So you only have to remember 2 of them, and then the other one is whatever is left, the other 2. Taking a closer look at the deoxyribose sugar. Now, one thing you have to know, like I had put before here on that deoxyribose, is the numbering system. You have the oxygen up there, then you start with the first OH. Okay, here in the case here is going to be a base, right? Okay, deoxyribose here is going to be the base, either purine or pyrimidine. So you have carbon 1, 2, 3, 4, and 5. I think everybody here took organic chemistry already, right? Or you're taking it now? No, it's not a requirement for this class. Well, this one doesn't take a lot of brain power. Just memorize, it goes around this way, clockwise. Okay, remember the way the clock goes? 1, 2, 3, 4, 5, and the 5 is the last one that is out of the ring, right there. 5 is not oxygen, talking about carbons here. So know how the carbon numbering goes. Now when you look at the deoxyribose, there you go, the guy is missing a foot in there. Okay, so that's my little person. Oh, missing the foot, that's the deoxyribose. 5 prime is right at the arm over there. Just to give you a little more details in the structures, which I said, you don't have to memorize. The purine base has this type of shape. 6 sides here, 5 sides there. It's nitrogenous base, it has nitrogen. Just adenine, guanine. And then if you have only 1 ring, it is the pyrimidine. And then you know that DNA will have thymine and cytosine, and then RNA will have uracil instead of the thymine. And that would be one of the differences between the DNA and the RNA. Okay, so we have already 2 differences down. First one is the sugar, that in RNA you have which sugar again? Ribose. In DNA, deoxyribose. Another difference, DNA will have thymine instead of uracil, or RNA will have uracil instead of DNA's thymine. Now, just looking at the molecule, it took me a long time to realize that ATP has something to do with RNA. It didn't really hit me until I looked at the structure of it. Now, we're not looking at ATP here, we're looking at AMP. Okay, what does that mean? It means if it is M, it has to do with mono, which is 1. P is a phosphate group right there. So if you have here, right, adenine here, deoxyadenosine, and you have only 1 phosphate over there, so there is no T here, it's a mono, right? Not triphosphate, it's a monophosphate. So if you read the name of the molecule, it should tell you what the molecule is. Okay. Now if you have 2 phosphates here, what's that going to be called? Diphosphate. And then if you have 3, it's going to be triphosphate. So this is just to give you just a whole picture of what those molecules look like. Okay. Now all the nucleotides in the DNA are connected via a phosphodiester bond. Now what that means? It means that on the phosphate, on the carbon 5, the one that is hanging out, right, the arm here, carbon 5 all the way to the other side, there is this phosphate group that connects to the next sugar. And it will connect to the 3' side of that sugar. That means on the carbon number 3. Okay, 1, 2, 3. So when you get this connection here, this nucleophilic attack with the dehydration, you get a phosphodiester bond. You'll know that you have two sugars connected by a phosphodiester, two esters. So again, you have a 3' one side will have a 3', 5' phosphate, okay, which is up there, and the other side we have a 3' carbon. You have a free OH group here on one side of the DNA, and then on the other side, after all these phosphodiesters bonds, you're going to have the 5' end, which is a free phosphate. So imagine this whole molecule, this whole chain of phosphodiesters. Then if you look at the very end, one of the ends is going to have a free phosphate, and all the way on the other hand, you're going to have a free OH group on the 3' carbon. Now, looking a little bit at the structure of DNA and how they figured out about the structure of DNA, before Watson and Crick, this was about 1 year before Watson and Crick, we had two scientists in the UK at the King's College that figured out what was the picture of DNA. So this was Rosalind Franklin and Maurice Wilkins. Now, nobody really talks about the Wilkins, Franklin, Watson, and Crick structure, but that's what they should talk about, because a lot of the work done was by Rosalind Franklin, actually. Actually at that time, woman scientists, not very common. So what happened here is that they crystallized DNA and decided to take an X-ray picture of it. Now, why do you think you need a crystallized structure to take an X-ray picture of it? Anybody took an X-ray to have a broken bone before?
Solid.
It has to be solid, yeah. Because when you take an X-ray, sometimes you have a broken bone and then they shift a little bit and the X-ray is all blurry. You can't see much, right? You can see the fracture. They say, keep still, hold it. Okay, I hold it. Okay, now you have a clear picture of the bone, solid and not moving around. In order to get an X-ray crystallography picture, you must have a crystal structure. You must have a solid structure. So they were able to crystallize the DNA, and then they used this X-ray source that shone, just radiated this crystal, and they used an X-ray film on the back just to capture whatever was coming through, and they came up with this picture here. So they figured out that it had to do with a helix, and they kept on getting very repetitive numbers out of these measurements. Now, the way they did these measurements, it was a very sophisticated way. They got a ruler, they went to the X-ray film, and they measure it one by one. Okay, so they realized there was some patterns that kept on coming up, like 3.4 or 34, and they realized there was something to do with maybe 3.4 as 1 unit and 34 as 10 units. So they were really close to what the structure of DNA should look like. But then they needed an extra leap to really figure out what it was. So this was 1952. And here, I couldn't resist. I put this couple of pictures that I took last year. I was actually in King's College last year. It was really cool. A little conference there. So coming back to the US here, 1953, there came Watson and Crick. And how did they figure this one out? They came up with all these ideas that, yes, DNA was a double helix, which means two of those phosphodiester bonded linked sugars, nucleotides had to be connected in two of them in order to represent this structure. Now, the way they did this was by using, as you see here in the picture, atomic models. They also had this very sophisticated equipment of little balls and little sticks. So you connect the sticks, you make the connections, you draw the molecule, you mount in 3D the molecule. Anybody ever play with that a Tinker Toy? Yeah, so you can come up with an extra credit for the structure of DNA out of Tinker Toy. It's really neat, because then you figure out what can happen. Okay? So they started to put all these molecules together, and they said, let's try to build it and see how we can associate what comes out of this to whatever is on that X-ray picture. Okay, so they put the molecules there, and they said, the distance between one side and the other of this helix was about 2 nanometers. And Crick was a chemist. he knew a lot about atoms and molecules. So he figured out that if the DNA helix was only one helix going around itself, it would be too small to be 2 nanometers in distance. If it was 3 molecules going around, because they thought maybe it could be 3, not 2, then it would be too big. Then they figured out it has to be 2, because that's the only thing it would fit into 2 nanometers of that distance. So they figured out, okay, it's 2 helix. Now how do they go together? So first thing they thought could be, okay, they all like pointing up. So they started to do the bases here. But then if you look at the base structure, now just look at it as a molecule, okay, as a Tinker Toy molecule. So now you have another sugar, another base. Let's see if the bases are going this way. Now there is a problem here because there's a huge bulk over here, and then at the bottom here you have something that goes tie skinny. So this is going to make you some helix that will look like that, in a zigzag line. And based on the X-ray pictures, this helix should be straight. So they knew that something was wrong with this type of setting, and they just worked at it as if it was a puzzle. Crick decided to invert this base. Now the sugar is on the bottom here. And now, yes, you have another base that will come and join somewhere here. You have, you know, all those hydrogen bonds here, right? And now the phosphate is over here. Now look what happens. If you pile up on top of each other these bases on this opposite way, on this anti-parallel way, then you have this straight looking helix. It doesn't look like a zigzag anymore. Okay, so playing with their molecules, that's how they figured out what the structure is supposed to look like. Okay, also, based on that, you look at this. If you have here purine and you have here another purine, that will also enlarge your base, enlarge your helix. So that was not compatible. And then if you have 2 of the pyrimidines here, that also shrink the base size, the helix size. So that was not compatible either. So based also on Chargaff's rule of the proportions of A to T's and C's to G's, they figure out, yes, of course, you know, A's go to T's, which is 1 ring, and then C will go to G. And then they still got the straight looking staircase of helix, right? So that's how they came up with that idea. Anti-parallel. Now the bases will lie flat in the middle. Okay, and then you have the carbon, sugar, the phosphate, and the carbon, they will go to the outside of the base. And also based on that structure, you know, remember that Crick was a chemist. They figured out that you have hydrogen bonds, 2 of them for A to T, and then 3 of them to C to G. No, you do not have to know exactly the structure, how that thing comes up together. Just remember how many there are. It's important for melting temperature. That's just another representation. Here is a 2 nanometer distance of a straight looking helix, straight looking double helix. Also, they figured out here that the 3.4 nanometer had to do here, 0.34 nanometer, it had to do with the distance between one base and the other, okay, one nucleotide and the other. And then the 3.4 difference had to do with this whole turn of this loop. So it would take 10 bases to do a whole turn of the loop. So which type of DNA were they working with there? Do you know? It's about 10 bases per turn. It's the most common form of DNA found. That's a bDNA, which you're going to see in a couple of minutes here. There we go. That's a bDNA, the most common form of DNA found. So that's what they were working with. But DNA does not only come in the B form. Okay, it can be an A form, and the way that you get this A form of DNA is by dehydrating the DNA. So you take some water out of the environment of the DNA, you shrink it a little bit, you see the coil here, the whole helix just shrinks a little bit. And then you can have another form of DNA that is called the zDNA form, that instead of being a right-handed helix, is a left-handed helix. And that occurs naturally. We just don't know where and how it works. But we know it occurs naturally because there are some specific proteins that will bind only to the Z form of the DNA, and not to the B form or the A form. So if you do have molecules that will recognize the Z form of DNA, you must have zDNA somewhere. You wouldn't have molecules that will bind something that doesn't exist, right? So you have an indirect evidence that zDNA exists. Also, the Z form of DNA is mainly composed of CG base pairs, okay, with those 3 hydrogen bonds, like very tight together. So once you get a lot of CG's sequences following each other, you get this helix that will turn the other way, and you have a form of the zDNA. Now, notice here that if you have the bDNA compared to the aDNA, you have the same right-handed helix, and the grooves are smaller. You see? The large groove here, okay, this big piece that you see like almost through the DNA, that's the large groove. Then you have the major groove here, right? And then you have the minor groove on the other side. And these shapes of the DNA are sites that can interact with proteins. Now, if a Z form, you don't have these grooves. See, it looks just like a string here. You don't have those grooves. In the A form, you also have the grooves. They're just compacted. They're smaller. They're dehydrated. Okay, so that's a little summary of what the DNA forms look like. Okay. Now, we know that aDNA is that dehydrated form of DNA. Now, where do you find it? Usually you find it in spores of bacteria, because when, spores of these bacteria that are exposed to the environment. So these bacteria are in a rough environment, usually without a lot of water, so they have to survive on a condition that is, dehydrating. So you can imagine here that the DNA is also going to suffer, it's going to lose some water, so it's going to be more compacted. It's going to become an A form of DNA. And also, when the DNA is more compacted, like it is here, you can imagine that it's harder for something to reach it. Okay? So if you're trying to shine UV light in there, chances are that you're probably not going to reach the core of the DNA very easily, because it's compacted and it's shrunk and it's in the middle. This is a way that these bacteria figured out how to survive in a rough environment for a longer time until they can find the next host to hop on. So these are the spores. Now, zDNA, as I mentioned, we know they exist, and we do know about proteins that bind to the zDNA. And we also think that close to the centromere, there are packs of zDNA structures, because those are very rich in CG bonds. You very compacted DNA there. Just a concept check. The antiparallel nature of the DNA is referring to what? The opposite direction. One string goes up, the other string goes down. Okay, one side goes up, the other side goes down. The opposite direction of the strands.